is associated with a disease called melioidosis. This pathogen is intracellular, but the bacteria can go inside, outside, move around. And for the United States, it's important because this is a select agent. So what that means is it can actually be weaponized and can be used as a biothreat agent, and has been actually used as a biothreat agent. So that's why in the United States there's a lot of interest about this bacteria. I do not develop weapons of mass destruction, so I'm interested in how these bacteria interact with cells. So if you put this bacteria in contact with phagocytic, non-phagocytic cells, the bacteria hangs around, survives, multiplies in macrophages, NK cells, dendritic cells, you name it. And the way you acquire this infection is 3 routes that we know. One is cutaneous infection through the skin. When you are in a rice field working, you can get an infection. If you're in a tsunami or in a hurricane, you can actually inhale it. Or if you are in Thailand and you're eating in the street in Thailand, you can actually get ingested the bacteria. The food is contaminated, the water is contaminated with it. Next. So, for many years we didn't know about the distribution of this bacteria. Everybody, when you hear talks about this, they say it's always located in Southeast Asia and Northern Australia. And we didn't know anything about it. So a survey was done about all the different countries that have actually reported cases of melioidosis. And as you can see clearly, the majority of the cases are located in Southeast Asia, India, Thailand, Myanmar, Laos, Cambodia, Northern Australia. But as you move on to the left in the screen, you can actually see that there's reports in Central Africa and a lot of reports in Latin America, and including Mexico. So, which was quite surprising that we actually have melioidosis in Mexico. And as you can see, it's a tropical disease. Basically, this is in the tropics across the globe. Next, please. One of my postdocs, Javier Sanchez, started collecting all the information about Latin America and figuring out what's going on. And as you can see, all the Caribbean islands have reported cases of melioidosis. All Central America has reported cases of melioidosis, and Mexico, they have reported at least 14 cases of melioidosis. Why do we have to be worried about this? Well, this is a lethal disease. Normally 50 to 70% of people die from this infection because it's misdiagnosed. In Mexico, we have the record. 85% of people infected with melioidosis die because physicians don't know how to identify this pathogen. Next. In Texas, we have had 2 native cases of melioidosis, but it has not been found in the soil in the area where normally you find this bacteria contaminated the soil. So we don't know why we have 2 cases in Texas. Next. Recently, you follow the news. There was this outbreak of this aromatherapy in the United States. People were infected when they were putting this aromatherapy at home. And they were getting infected with this bacteria. It came from a producer in India, and we got infected. Next. And in the United States, finally, this infection became endemic. So what that means is actually they isolate the pathogen from the soil of a location where people were getting infected. And in Mississippi, we have at least 3 cases of melioidosis, and they were isolated. Here in Mexico, if you are curious, if you Google Mexico melioidosis, the last 5 cases that we got was in an area of Mexico where a lot of tourists go. It's not Cancun, it's La Paz, Baja California. For 4 people died from melioidosis. Next. So, as I was telling you, in Mexico we have an issue because this was after a hurricane. And 5 people from the area got infected, 4 of them died. And the problem is they couldn't figure out what the pathogen was until the people was there. They sent the samples to the INTRA in Mexico, which is the reference laboratory, and they finally tell them, oh, you have Burkholderia pseudomallei in Mexico. And this is concerning. And the other thing that is happening in Mexico is all this migration coming from South America to Central America to Mexico. A lot of these kids that through the Darien jungle, they're getting infected, they come to Mexico and they die from melioidosis. Those are not reported. There's a lot of cases that are not reported. Next. So again, This bacteria, once you acquire it, the bacteria can go anywhere. And you can get infections almost everywhere. So the bacteria can go to the respiratory tract, you get a pneumonia kind of infection, urinary tract infections, it can go to the brain, you can get in the gastrointestinal tract, muscles, skin, you name it. The bacteria, nothing can stop this bacteria. Next. So we are going to be focusing, since the talks that we have is gastrointestinal tract, we're going to be talking about gastrointestinal infections. Next. So what Dr. Finley was indicating is my lab has been interested to understand how this bacteria cause infection. So we ask very simple questions. If we have a model in the laboratory with intestinal epithelial cells, can the bacteria enter the intestinal epithelial cells? And what happens once the bacteria enters these intestinal epithelial cells? And to ask the question, we have a needle-like structure called the type 6 secretion system that Dr. Finley already explained to you. This is not outside the bacteria. This is a needle that shoots. Every time the bacteria is needed, it shoots this bacteria. And so far, we think there is one effector protein. I was presented type 3, you have a lot of effector proteins. In Burkholderia, we only have one. And Burkholderia has a lot of everything. So if EHEC or EPEC has one type 3, we have 3. If EHEC and EPEC has one type 6 secretion system, this organism has 5 systems. We only work with one, called the type 6 secretion system 1. And we asked the question, so we knew that once you have this system inactivated, or a mutation, the bacteria is unable to move from cell to cell. And the type 6 secretion is essential for this movement. And this bacteria is very smart. Once the bacteria goes inside the cells, the bacteria doesn't need to go outside. It fuses the cells and forms a multinucleate giant cell and moves around, survives, uses the nutrients, and goes around and infects other cells. And it requires a type 6 secretion system. Next. So, since we're in a cell biology, microbiology system, we always have to have nice pictures. So this is Burkholderia, that the Walter strain that you can actually see with time. The bacteria start multiplying. You can start seeing actin tails that allows the bacteria to move around. So they multiply. They have these beautiful actin tails. And the speed is, this bacteria moves really fast. At the speed of Legionella, Listeria, or Shigella, it really moves really fast inside the cells. Next. So the bacteria adheres, invades, disrupts the cells, and forms these multinucleate giant cells. You can actually see the nuclei of the bacteria all together. They are not disrupted, the membranes are intact. Next. And with the type 6 secretion system mutant, you can still find bacteria, but then the bacteria start multiplying, but the bacteria is trapped inside the cells, and very few actin tails are formed after 24 hours. And there is no multinucleate giant cells. Next. So, this is essential next for invasion. So, since we have a model, so we can interrogate, once the bacteria is inside, you have this multinucleate giant cell, and eventually the cells are going to die. Can we actually ask the question, can we actually determine whether the bacteria is moving and killing the cells? So, for those that work with viruses, there's something called plaque assays, so you can see cells infected with bacteria or viruses. And if the virus is killing the cells, you have a hole. And you can stain these, and you have these plaques. Burkholderia works really beautifully, doing exactly the same. You can actually have plaque formation, and then you can actually do it with intestinal epithelial cells or any other cells. So we ask the question, can we identify potential virulence factors that are associated with this dissemination? Next. So I told you, if you inactivate the type 6 secretion system, you inactivate invasion, you don't have plaque formation. Next. Something that was surprising to us, if you inactivate the type 1 fibria, the bacteria is unable to form plaques or reduce plaques because the adhesion is reduced. And we already showed that. I don't know why the bacteria requires that. But we also, in my laboratory, we also develop animal models, and everything that we do in vitro goes in vivo. So we ask whether this bacteria can be used to infect an animal model, in two different ways, an acute way or a chronic infection. So in the acute way, we actually use a larger dose, LD50, and the bacteria basically kills the animals within 21 days. Or if the mutants that we're screening, they might be able to survive at 21 days. And with a lower dose, which is 1 LD50, you get animals that survive to 35 days, and then the pathogen can disseminate to internal organs, and we actually quantify this dissemination of what organs are infected. Next. So in the acute model, we have the type 6 secretion system. We compare it with the type 1 fimbria and the double mutants. And as you can see, attenuation with the type 6 secretion system mutant. It doesn't matter if you have type 1 fimbria, you still have killing of the animals. And then you basically see that the animals colonize very well, are colonized in the different organs, stomach, small intestine, colon, and cecum. We basically do not found bacteria in the stomach and colon, because the bacteria disseminates really fast. If it's the wild type, the bacteria doesn't stay there, it moves really fast to other organs or kills the animals. Next. But when you look at the chronic stage, when you use a lower dose, the wild type still kill the animals, but the mutants are attenuated, and you can actually see different patterns of bacteria colonizing different organs. You start seeing now that the wild type start appearing in the spleen, and eventually, if we leave this longer, you start seeing the bacteria going to the liver, and if you take any organ that you are interested, you can actually have it. Next. So, using this model, we have already the system where we can identify. We have the system to screen it. We have the animal model. Next. We ask the question, what other virulence factors are important? So, next. So we did a dual RNA-seq analysis where we actually asked the question what genes are upregulated or downregulated in the host, what genes are upregulated or downregulated in the bacteria while the bacteria is infected. And since we have the type 6 secretion mutant, we can compare the wild type versus the type 6, and you can have a time course comparing the two of them. So you have to be proud, two Mexicans did this work, Javier and Daniel. Next. So obviously, I'm sure you will not be able to see this. It's just that it's a volcano plot just to show you that some of the genes are upregulated, some of the genes are downregulated. We have a transposon library that in every single open reading frame of Burkholderia pseudomallei, it's about 5,000 genes in the 2 chromosomes of this bacteria. So we actually pull out these transposons and start interrogating which of these mutants that are upregulated might be associated with plaque formation. As you can see, most of them form plaques. But then we found some, this is the wild type with the plaques, this is the mock, and then we found one that was unable to form plaques. It's called BK. So we decided to look at BK, what the role of BK was doing. Next. No, the other way. Yeah, one more. Keep on. Oh, you're going backwards.
What else?
Yeah, you got it. So, we found out that BK, together with another protein called ESAN, somehow are associated with regulation of secretion systems in Burkholderia. So, somehow affect type 3 secretion effectors as well as type 6 secretion effectors, and also somehow it's associated with motility. So we asked the question, can we actually mutate BK, complement it, and see intracellular replication? As you can see, not surprising, wild type start multiplying very well, the complement the strength the same, and then the BK mutant has a defect in intracellular replication in these cells. So somehow this is regulating the 2 type 3 and type 6 secretion system. Next. So as you can see, we always look for multinuclear giant cell formation. You can actually see the wild type forming these beautiful multinuclear giant cells, a lot of nuclei and cytoplasm intact. The BK mutant is unable to form fusion. You can still see bacteria inside the cells, but you don't see this formation. And then
In the complemented strain, you see again the formation of these giant multinuclear cells, a lot of bacteria, and you start seeing actin tails in each one of these bacteria. Next. So, as I told you, we always tested in the animal model. The BK mutant is attenuated compared with the wild type in the acute as well as the chronic model. And when we start seeing for distribution, you can see that the BK mutant somehow is trapped in the colon, cecum, and spleen. It doesn't disappear, it's there, but it's unable to disseminate. And when we look for dissemination, you can clearly see that the wild type and the complemented start disseminating. The BK mutant is unable to disseminate. Next. So that's the first part of the story with this regulator that we think is important for these secretion systems. But as I told you, the bacteria is a system where once the bacteria is inside the cells, there's a lot of inflammation, a lot of rearrangements, but the bacteria is able to move from cell to cell to escape potential changes in the cell and allows the bacteria to move from cell to cell. But also another important component next of this bacteria is the bacteria doesn't stay in one organ, disseminates. And the way the bacteria disseminates is using macrophages. So the bacteria infects macrophages, survive inside macrophages, and the macrophages work as a Trojan horse to move it to different organs. Next. So one people in my lab, Joseph, was interested to understand whether macrophages were playing a role. And to our surprise, a lot of intracellular pathogens that we know, or some different pathogens that use macrophages for dissemination, they either use polarization of these macrophages to do their role, the pathogens. For example, M1, Shigella, Rickettsia, Francisella, they use M1, so pro-inflammatory macrophages. Other pathogens like Chlamydia, Yersinia ehrlichia use M2 polarization, so those healing macrophages. And to our surprise, Burkholderia, we have no idea whether they were able to polarize cells or not. So we started interrogating the pathogen to try to figure out whether the pathogen was able to mobilize one way or the other the polarization of the macrophages. Next. So Jacob in the lab started asking the question, can we use raw cells or bone marrow-derived macrophages? And again, very simple questions. The BK mutant, they complemented, what happened to the cells? And it's not surprising, BK mutant is destroyed in raw macrophages as well as bone marrow-derived macrophages. Wild type and the complemented strain, they multiply very well in macrophages. Next. So he asked the question, what happened when you have this mutant in macrophages? What happened to the type 3, to the type 6, and the actin motility? And this plot is just representing 2 time points, 3 hours and 6 hours. And the colors indicates when basically the genes do not change, they are in white. When you have slightly changes in green, and when you have upregulation of these genes in red. And as you can see with time, with this BK mutant, somehow the type 2 secretion effector proteins are upregulated, a little bit of the type 6, but the other ones do not really change. And as I told you, just to remind you, BK seems to be a regulator controlling somehow type 3 effector proteins, but also type 6 secretion system and active motility. Next. So we test this system. We moved from the gastrointestinal tract to use a model where we can actually test more systemic infection. So we use the inhalational model of infection. So we put the bacteria in the nostrils of the animals, and we ask the question, what happened in the lungs, and whether these animals can survive. So you can see that the wild type and the complemented killed the animals within 4 days. The BK mutant survived very well. And this is just the body weight you can actually see. And when you start looking at the different organs, you can see dissemination in the different organs using this route of infection. Next. So why do we use the intranasal route of infection? Because we can actually take a time point, 48 hours, and when we still have animals surviving, and we can actually take macrophages from the lungs and then ask the question, in the lung, what happened with the macrophages? Next. And again, I put flow because they eat it. I do not understand. To me, they're all dots. But basically what we do is you gate it, looking for different macrophages, and you have markers for N1 or N2 macrophages. This is too small, so I'm going to jump to the next slide, where we actually, it's the same pictures and the same graphs, but you should represent it. When you look for total macrophages, there is no significant difference between the PK, the complemented, or the one. Total macrophages population is the same. Next. When we look for N1 macrophages, you start seeing reduction in macrophages if you look for total cells. But you do for proportion of total macrophages, there is no significant difference in N1 macrophages. So there is no effect in pro-inflammatory macrophages when you have a PK mutant. So this is regulated. But what happened with the N2 macrophages? With the N2 macrophages, basically you abolish the activation of polarization to N2 macrophages. So somehow this regulator, and either type 3 or type 6, somehow are involved in polarization of these macrophages. So you might ask the question, well, you already have the mutant with the type 6, why you didn't test it? Well, next. Well, this is a PK dependent into polarization. We ask the question, type 6 secretion is important in polarization. The long story short, type 6 secretion does not participate in polarization. So it's not. So we have two options now. Obviously, that's what we're investigating. It's a type 3 dependent mechanism, or there's something else that PK is controlling. However, this mutant somehow controls apoptosis. So type 6 secretion is participating in apoptosis, is not participating in polarization of the macrophages. Next. And what we have found is definitely using flow, you can have exacerbation of apoptosis in macrophages during infection when you have the HCP mutant. So you have the wild type HCP mutant complemented, and you look for percentage apoptosis in cells, and you can actually see that the wild type and the complemented exacerbate the apoptotic events compared with the HCP mutant. Next. So, just to tell you, I walked you through a lot of cell biology, molecular biology, pathogenesis, virulence factors. We did a dual RNA-seq, so we know that type 6 secretion is important. We know that type 3 and type 6 are connected in this pathogen, at least one of the type 3 and one of the type 6. I don't know what the other 4 type 6 are doing. I have no idea what the other 2 type 3 secretion systems are doing in this bacteria. We already showed you that it's attenuated. We showed you about N2 macrophage polarization. So the question is, why do you need this polarization? What is this bacteria doing with polarization? And we're also asking the question, what is this exacerbation of apoptotic cell death? As Brett was indicating, apoptosis is an issue when you have an extracellular pathogen. You don't want to exacerbate the apoptotic pathways because you can get killed. But this bacteria seems to like that, because maybe the bacteria use this inflammatory response to move around and escape. So that's the question. Next. I think I finished on time. So this is currently my lab members, and most of the work I represented. Jacob has done all the apoptotic things, and Sarah and the other ones are working with vaccine development. And thank you very much for the attention.